Hello, good evening everyone. Welcome to Easy Nurse, where nursing made easy. Let us start our live daily dose session. So, with regarding to applied biochemistry, which is applicable for PBBSc students and also BSc student and GNM student. So read carefully and listen carefully. This is very important for, and this is very applied biochemistry is very important for the future. So in this session we will discuss about the unit 3 enzymes. What is enzyme and what are the factors that is affecting the enzyme, and also what is the mechanism that is involved in the enzyme activity, and also we'll discuss about the diagnostic application which is applicable, and also we'll discuss about the digestion and absorption of the carbohydrates. Without wasting time, we'll just discuss about the definition of enzyme. So what do you mean by enzyme? Enzymes are nothing but they are the biocatalyst. Biocatalyst. In the name itself, it is saying that is bio means living. What is that? Bio means living. And catalyst. What do you mean by catalyst? Catalyst is nothing but where the reactions is catalyzing. Okay, that is biocatalyst. Biocatalyst is where you synthesized, it is synthesized in the living tissues. Where it is synthesizing? It is synthesized in the living tissues, and which rates the or which speeds up the rate of reaction without consuming. Okay, in the process the enzymes, that maybe it is a structure or anything, it is not consumed. Okay, so enzymes is defined as a biocatalyst which is synthesized by a living tissues and which speeds up the rate of reaction and which is not altered or which is not getting altered within the during the process. Okay, for example, some reaction is taking place in our body, but in that the enzyme which is very responsible to complete that reaction, in that reaction the enzyme which will not consumed or which will not altered its shape and its size. Okay, so why it is very important, why you have to study, basically why the nursing students wants to study this enzyme, because these enzymes which will accelerate, okay, which will accelerate the vital means very important chemical reactions such as digestion. Okay, where it is important? Because it is for the digestion purpose it is very important, and for the metabolism. Okay, where it is metabolism, and also DNA replication. For the DNA replication, digestion, metabolism, for all this the enzyme is a crucial or it is very important. And next, what are the examples you can see in the enzymes? One is salivary amylase. Where it is present? It is present in the saliva. And lipase enzyme. These lipase enzyme which is very important for the breakdown of the lipid components. And proteases. Proteases is an enzyme which is very important for the breakdown of protein molecules. And DNases, like for nucleotides. Okay, for the nucleotides breakdown you need DNases. Okay, these are the example for enzyme. And also we'll see some important terminologies. For example, that is cofactor. Okay, cofactor is one of the important terminology in the enzyme concept. Okay, what do you mean by this cofactor? Cofactor is nothing but it is a non-proteinous part. What is this? It is the non-proteinous part. Okay, this is the non-proteinous part, and which is divided into two groups, that is coenzyme. Okay, Coenzyme. Coenzyme means it is also a non-proteinous part, but which includes the organic compounds. Okay, which includes the organic compounds. For example, what are those organic compounds? NAD plus, okay, NAD plus and FAD. These are the organic compounds. So these are the main terminologies, that is cofactor and coenzyme. Both are same, but here it is a non-proteinous. In here also coenzyme also it is a non-proteinous, but it is a organic compound. It includes the organic compounds. But here in the cofactor it includes the inorganic compounds like Na plus, okay, Mg2 plus. These are the cofactor. Okay, Na plus, Mg2 plus, these are the cofactors and coenzyme. And also one of the main important terminology is enzymes active site. Okay, enzymes active site. What do you mean by enzymes active site? You know that the structure of the enzyme, consider this is the structure of enzyme. Okay, this is the structure of enzyme where exactly the substrate, okay, where exactly the substrate which binds to the active site, okay, the place where exactly the substrate will bind and forms a reaction, that is called active site. The region, you can say, or you can define the active site like this: the region where exactly the substrate will bind and makes the reaction, that is from the reactant to product, which completes the reaction, that you known as active site. So this is nothing but it is a small pocket like structure or a groove like structure which is binded, okay, which binds to the that is where the substrate will bind to the active sites. Okay, this is about active site. And also enzyme activity, this is also very important in the enzymes concept, that is enzyme activity. What do you mean by enzyme activity? You know that how much, okay, the measurement of the reaction, okay, for example, the reactant A and which converts into a reactant B, or the product, okay, the reactant which binds to the enzymes and forms a product, at what site or at what measure, okay, that measurement you call it as enzyme activity. You can define as the measurement how much reactions the enzyme can catalyze, or the catalysis of the enzymes, that you call it as enzyme activity. Okay, so next we'll see what are the classification of enzyme, okay, according to IUBMB. According to IUBMB, what is the abbreviation of IUBMB? International Union of Biochemistry and also Microbiology. According to them, the enzymes which are classified into 6 groups. How many groups? It is 6 groups. Okay, first one, oxidoreductase. What is that? Oxidoreductase. In the name itself, it is telling oxidation as well as reduction in the chemical reaction where the oxidation as well as reduction takes place. That process is called as oxidoreductase. The enzyme which catalyzing the reaction both, that is oxidation as well as reduction, that you call it as oxidoreductases. This is the enzyme. And keep it in mind, all the enzymes which is end with the synonym, that is ases, okay, that is suffix ases, it ends with the suffix ases, that is reductases. You can observe here reductases, transferases, hydrolases, ligases, okay, it always ends with the ASES. Fine. Next, oxidoreductase, example, dehydrogenase. This is also one of the important enzyme, dehydrogenase. And LDH. LDH means lactate dehydrogenase. As well as you have catalase enzyme. oxidase enzyme, which catalyzes the oxidation reaction, and reductase enzyme, which catalyzes the reaction reductase or reduction. Next class, that is transferases. What is that? Transferases. What do you mean by transferases? Here also you can observe transfer. The name it's suggesting transfer. What it is transfer? Here the functional group, okay, from the molecule, from one molecule to another molecule, or one compound to another compound, the functional group is transfer. Okay, the transfer of functional group from one molecule to another molecule, that is catalyzed by the enzyme called transferases. For example, you can observe in the glucose you have CHO. Okay, the group is CHO, the aldehyde group, and which will be converted into keto group, that is C double bond O. When it is converting into fructose, from glucose to fructose, there the reaction, that is the functional group, is changing. Okay, or transaminase, you can observe that transaminase, the amino group which will be transferring from one molecule to another molecule. Which enzyme it is catalyzing that reaction? That reaction is catalyzed by transferase enzyme. For example, transaminase. What is that enzyme? Transaminase. Here the amino group, okay, the amino group will be transferred. And PFK. What do you mean by PFK? Phosphofructokinase. What is that? Phosphofructokinase. This is an enzyme which catalyzes the transfer of functional group, and also kinase enzyme, and many more enzymes are present under the transferases. Okay, next is hydrolases. Hydrolases. Here also the name suggests hydra means what? Water. Using the water molecules, okay, using the water molecules, the breakdown, okay, what is here? Here the bond is breaking down, okay, between the molecules there will be some bond, okay, that bond which will be break down by the enzyme called hydrolases. Okay, the hydrolases is an enzyme which catalyzes the reaction using the water molecule. It breaks down the bond. Okay, example, that is maltase. Okay, this is maltase is maltose, the maltose sugar which will be broken down the bond by the enzyme called maltase. And sucrase. Sucrase, the sucrose, the sucrose compound is broken down by sucrase enzyme. And amylase, amylase enzyme, lipase, these all are the example for hydrolases. Next, lyases. What do you mean by lyases? Here also same, lyase or lyse, you have heard this, lysing or lyase. These lyases enzyme which catalyzes the breaking of bond. Here also the breakdown of bond takes place. Okay, breakdown of bond, but without using water. You can observe, don't confuse here with the hydrolases and lyases. Both the action is same. Here also bond is breaking and here also the bond is breaking. But the difference is, in the hydrolases the water molecule is used up, but here there is no water molecule is used up. But by the help of energy only it will break down the bonds. For example, aldolases, aldolase enzyme, fumarase enzyme, and decarboxylase. Decarboxylase means which removes the carbon dioxide, CO2 molecules from the molecule. These all are the example for lyases enzyme. And isomerases. What do you mean by isomerases? Isomerases means it is shifting the molecules. Okay, it's shifting the hydrocarbons, in changing the carbon atoms, that is changing, that is isomers. Okay, for example, epimerase enzyme, mutase enzyme, and isomerase enzyme. Here also in the changing of glucose to fructose. Okay, you can, if you know the structure of glucose and fructose, you will get to know. At the first position of the glucose you have the CHO, and in the fructose you have the CH2OH. So the hydrocarbons and the carbon atoms which will be isomers, that is catalyzed by isomerase enzyme. Next is ligases enzyme. What do you mean by ligases? Ligases is nothing but which helps to join, okay? When, for example, there is some DNA which is broken, okay? That broken DNA which will be ligated or joined by the help of an enzyme called ligases. Ligases is nothing but which is helps to join the molecules, okay? For example, DNase enzyme and pyruvate carboxylase enzyme and also glutathione synthetase enzyme. These are the enzymes which is very useful for the ligation. Ligation means joining of the compounds or an molecule. And this is the sixth classification which was given by IUBMB, that is International Union of Biochemistry and Molecular Biology. Next, let us discuss about the mechanism, how the enzyme activity is measured and also how it is how it is mechanized, okay? There are two theories. Actually, there is two theories. One is lock and key model and another one is Koshland's theory. First, we'll see the structure of an enzyme. Consider this is an enzyme structure as we have discussed earlier, and this is the active site, okay? This is called active site, and you have a suitable substrate, okay? You have a suitable substrate, and when this substrate it's bind to the active site of an enzyme, this forms a transition state that you call enzyme substrate complex. What is that? Enzyme substrate complex. This transition state you call it as enzyme substrate complex, and then it will form after the reaction. It gives the enzyme plus product. This you call it as reactant, and when it complete reaction occurs, then it gives the enzyme and product separately, okay? This is how the enzyme is mechanized, okay, how it works. Then coming to the Fisher's template theory, this is helpful for the how the active site is formed and also how the substrate will bind. Everything we'll see in this Fisher template theory. This Fisher template theory is also known as lock and key model. Why it is called lock and key model, you will get to know now. So this Fisher theory was given by Emil Fisher, okay, in 1895. Okay, in 1895, the Emil Fisher is a scientist who introduced the lock and key model. We know that lock and key concept, right? For the particular lock, we have a particular key. Likewise, the scientist Emil Fisher introduced that, or he told that there is a concept that is for particular active site, there will be a particular substrate. For example, for particular enzyme, there will be a particular substrate which will binds together. For you can observe here, this lock has key which will binds and opens the lock, and here also you can see. Likewise, in the enzyme, you have an enzyme and shape which will binds the substrate and forms the enzyme substrate complex. And look at here. So I told that active site, right? This is the active site. See the shape of the active site. Here the shape of the active site is different, and here also the site of the shape of the active site is different. So the substrate should be same size of the active site. Okay, the active site shape and the substrate binding shape, it should be equal and it should be similar. Then only the enzyme and the substrate will bind. Otherwise, it won't bind and the chemical reaction will stop. If it alters the substrate shape and if it is alters the active site shape, that is the chemical reaction will be stop over there. Okay, then here you can observe that the active site will be rigid. What is there? The active site shape will be rigid in nature. You cannot alter here. You cannot alter, and this the substrate which binds to the active sites and form a enzyme substrate complex. And this theory is not acceptable. Why? Because this is depends on the shape of the active site. That is the reason the Emile Fisher cannot explain the structure of the active site and also its mechanism properly. That's why this lock and key model, according to regarding this enzyme concept, it is failed. What it is? It is failed and it is not acceptable. So after this, the Koshland, okay, who is that? The scientist called Daniel Koshland. Okay, Daniel Koshland, who introduced the theory called induced fit theory. What is that theory called? Induced fit theory. He taken reference of lock and key model and he introduced himself in induced fit theory. According to this theory, it says the active site, okay, the active site of an enzyme which will be altered based on the substrate size. Okay, you can see, you can observe here the shape of the substrate is different and the active site of the enzyme is also different, and here the slope, the bell curve shape of the substrate, but here it is in like slightly rectangle, and here the shape of the active site is triangle. Here it can fit, but here it cannot fit according to the lock and key model. But he told that, who told? The Koshland scientist told that the active site of an enzyme can be altered or it can be changed according to this substrate. Okay, according to this substrate it can be changed, and this creates the enzyme substrate complex. And here the active site will be flexible. Okay, what it is here? Flexible. But there lock and key model, it is rigid, but here the active site will be flexible. Understood? So according to the Koshland theory, it is acceptable because all the enzymes and the substrate which will according to the lock and key model, only for particular enzyme there will be a particular substrate, then only it will forms a reaction. But here the enzyme's active sites itself, it will changes or alter and according to the substrate and forms a reaction, that is enzyme substrate complex. This is what the mechanism, how it works, the enzyme's activity, and how the substrate binds to the enzyme active site. So next we'll discuss about what are the factors that is affecting the enzyme activity. Understood? So there are many factors which affects the enzyme activity as we have to study. For example, as we have to study, we don't want any distractions, right? For example, if we want to study biochemistry, what we need? We need some biochemistry textbook and some reference books, running notes, etc. We need. Likewise, for enzymes also, there is some factors. At that time only it will activate. Otherwise, it will be in the inactive state. What is that? Inactive. If the factors, okay, if the factors are not favorable, at that time the enzyme will be inactive state. When the favorable factors or the favorable conditions is applicable, then the enzymes will be in the active state. Okay, remember this. Then coming to the factors, we have enzyme concentration, and we'll discuss the temperature and time kinetics, how this factor affects the enzyme activity, and pH, very important, pH and substrate concentration. These are the 5 important factors which actually affects the enzyme activity. First, we'll see the enzyme concentration. What do you mean by enzyme concentration? When the enzymes is adding up to the reaction, okay, you have for example, you have 80%, okay, but you have substrate like 40% or 60%, Okay, you have 60% of substrate and you have a enzyme concentration is 80%, or you have equal 40% of enzymes and 40% of substrate. When you're adding the enzyme concentration, when the enzyme concentration increases, the substrate and the activity, okay, the activity of the enzyme will also be increases. Okay, here the enzyme activity is dependent upon the enzyme concentration. If you are providing more enzyme, the activity is also it's more. So this states that when the enzyme concentration is increases, the activity of an enzymes is also increases. When it stops, okay, at some point or at some condition, it will stop, it will be saturated and gives the linear curve. Okay, this graph is very important. You have to mention this graph while describing in the test or exam. So enzyme activity depends on the enzyme concentration. When the enzyme concentration is increases, the activity of an enzyme is also increases. Next, we'll see the temperature. Why temperature is very important? We know that for each and everything we have a temperature. For example, our body temperature is different and the surrounding, that is room temperature, is also different. For example, room temperature may be 27 degrees Celsius or 26 degrees Celsius. Okay, but our human body temperature is about 36 degrees Celsius. This is the normal temperature. So the enzymes which will be affected, okay, beyond the temperature and above the temperature, if it goes, the human enzymes will be inactivated stage, or you can say denature. So when the enzymes, okay, when the temperature will be increases, when the temperature will be increases, the enzymes activity is also getting increased at particular temperature. For example, our body temperature is 36 degrees Celsius. The enzymes which is present in our body, that should be in this condition. Okay, and also there is called optimum temperature. Optimum temperature means at maximum level, the temperature where the activity of an enzyme which reaches at a specific point, that you call it as optimum temperature, and after that it will falls down here. You can see the bell-shaped curve. Okay, this is called bell-shaped curve, where the molecules will be moving faster and more collisions occurring. So this optimum temperature is very important to maintain the enzyme activity. So there is a particular temperature for different enzyme. For example, for salivary amylase, there is a particular enzyme, and lipase, there is a particular temperature. At that temperature only, the enzymes will be activated. If the enzymes in beyond or above that, the enzymes will be denatures or inactivated. That's why the curve you can see here. Here only the maximum activity it shows, but here and between this, the activity will be less or denature. Okay, this is about the temperature. And then time kinetics. Coming to the time kinetics, here also initial stages the activity will be high, or in the initial stages the enzyme activity will be started, and at the optimum level, this is called optimum level of the time, at the particular time. For example, we are also having some saturated points. If someone is triggering us, we will just blast out, right? Likewise, here also there is a time for enzyme activity where exactly it activates and also inactivates. When it inactivates, when the time is not favorable for that particular enzyme, at that time it will inactivate. Okay, when it is favorable, it will be activate. That you call it as optimum time. Understood? This is what time kinetics. And next will be optimum pH. What do you mean by optimum pH? Optimum pH is also, you know that pH, for example, salivary amylase, we are discussing as far salivary amylase, that the pH of salivary amylase is 7. Okay, and next is pepsin. For example, pepsin, this is about 2. Okay, it is produced in the stomach, that is why it is highly acidic in condition. So in the salivary amylase which is present in the mouth, that is neutral. So this is about pH. Okay, when the pH, the enzyme concentration in the pH, the enzyme activity will be particular pH. As I told, for salivary amylase 7, only at that pH only it will be activated. And pepsin, it is acidic in condition 2 or 1.8, like that it will be present. So at that condition only, in that particular pH only, the enzyme will present. So just think that, or just remember, different enzyme has different kinetics. Okay, for example, salivary amylase 7 and 36 degrees Celsius, like this, temperature, pH, for different enzyme, different pH kinetics will be there. This is what, and above and below that, here also same, the enzyme activity will be inactivated or denatured. This is about the pH condition. And last one, that is substrate concentration. What do you mean by substrate concentration? Here also we know that in the enzyme you have active site. Okay, if the more enzymes are present, there will be a more active site. Okay, and also if you are providing more substrate for one active site, what happens? When one substrate binds, it will satisfy and it creates the, or it completes the reaction. Or you're uploading or you're creating more substrate, then that substrate will lead to saturation. Okay, when you're providing more substrate, it will binds to the complete set of active site and it satisfied. Once it's satisfied or once it is saturated, it gives a linear curve. Okay, this is what substrate concentration. So this is all about how the enzyme activity is affected by so many factors. Next, we'll discuss about the diagnostic application of enzyme. Why it is important? You all got some questions, right? Why it is important? Why nursing students should study these enzymes? What is the importance? Because this is very important for the diagnostic application. Like you have to diagnose something or some organs. How will you diagnose? By the help of these enzymes, you can diagnose the proper functioning of the organ. For example, cardiac function assessment. For the assessment of the cardiac system, to check the proper function of the heart. Cardiac means heart. The cardiac system, for the heart function, whether it is performing properly or not, to check that you need enzyme. What are those enzymes? You will check here the increased level of aspartate amino transferase. In short, you will say AST. This is the enzyme which is helpful for the cardiac function. And also creatine kinase. Okay, this is also the creatine kinase, that is CKMB. This is the isoenzyme. What do you call this? Isoenzyme. Okay, this is isoenzyme. And also you can check in the serum, that is used to detect the myocardial infarction, that is heart attack. To check the heart attack, to assess the heart attack, you need some enzyme, that is CKMB and also AST. These are the enzymes which is useful to assess the function of cardiac, that is cardiac system. Next, liver function test, that is LFTs. You have observed this is LFTs. While when you went for hospitals, they give some summaries, right, or prescription to check the liver function test, whether your liver is performing properly or not. What is the function of liver? It is detoxifies the drugs and other molecules, right? Mainly it is for the detoxification. And how will you check the liver is properly function or not? That is serum alanine transaminase, that is ALT. This is an enzyme which helpful to assess the liver function, and also which is the liver marker. It is a marker for the liver, and which is disease for the particular disease, and also hepatitis and toxic liver necrosis. For all this to check or to assess, you need enzyme. Next, bone hand hepatobiliary condition, for the assessment of the bone whether it is performing properly or not. Elevated means which is for example the levels will be alkaline phosphatase, the enzyme called alkaline phosphatase, which is used to check or which is the marker for the bone diseases like rickets. You have seen this, rickets, and also liver bile obstructions. Okay, for the liver bile obstructions, and also to check the bone, that is this ALT, which is act as a marker. And next is pancreatic disorders. What do you mean by pancreatic disorders? Which is destruction of the pancreas, or which is the ability which loses to produce some enzymes. That is how you check, by high serum lipase and also amylase. These are the levels. If you check the levels of the amylase and lipase enzymes, you can detect the pancreatic disorders. And also if you have acute pancreatic and chronic pancreatic. Next, prostate and other conditions, like what is that? Acid phosphatase. This is also very important enzyme which is helps to assess the prostate cancer and prostate issues. Regarding to prostate, you can check using the acid phosphatase. So these are the diagnostic applications for enzymes. And we all see this is as for now we have discussed all the aspects. Now the main important thing is digestion and absorption of the carbohydrates. As I told earlier, these enzymes which is very important in the digestion, metabolism and DNA replication. First we'll see the digestion and absorption of carbohydrates. What do you mean by carbohydrates? First of all, carbohydrates are nothing but they are the saccharides. What are those? They are the saccharides. You call it as saccharides. That is nothing but sugar units. Okay, sugar units, or it is a simpler sugar. It is a dietary, and these carbohydrates which provides the main source of energy. What it provides? It provides main source of energy. If you are intaking the carbohydrates, you will get the main source of energy, that is 4 kilocalories per gram. 4 kilocalories per gram. In 1 gram of carbohydrates, you will get 4 kilocalories. This is the main energy source by which you will get from carbohydrates. Let us discuss now how the digestion, if you are intaking the carbohydrates, how the digestion takes place in our body, that is in the human body, and how it will be absorbed. Okay, what do you mean by food? Food is nothing but which can be eatable and drinkable, that you call it as food. So sources of carbohydrates. First of all, we know that sources of carbohydrate. What are the sources of carbohydrates? Vegetables, corn, okay, corn, vegetables, fruits, grapes, in the fruits, grapes, and rice, right, and wheat. These are the sources of carbohydrates. And almost what we are intaking, that is in the form of polysaccharide. Okay, in the form of polysaccharides, we are intaking the carbohydrates. Those carbohydrates which is digested by two mechanisms. Okay, when you intake the food, for example, here you are intaking the rice or chapati. Okay, take it an example of rice. You are intaking the rice. When you intake into the mouth, that is called mechanical digestion. The first step is, what is that? Mechanical digestion. Mechanical digestion. What do you mean by mechanical digestion? Using the buccal cavity. You know that buccal cavity, what? The tooth are present, okay? Using the tooth, the food molecules which is intaken, that will be grinded. That is called chewing. What is that? Chewing. Or this process, the grinding process, you call it as mastication. What is that process called? Mastication. Okay, this mastication is nothing but chewing or grinding the food particles. When the grinding takes place or the mastication takes place, the saliva which is present inside the mouth, it comes into the contact with the food, and in the saliva there is an enzyme called salivary amylase. What is that? Salivary alpha amylase. Okay, salivary alpha amylase, which is present below the tongue. Okay, there will be a salivary gland below the tongue. Then that salivary gland which produces the salivary amylase enzyme, that salivary amylase enzyme which initiates the digestion in the mouth. Okay, which initiates the digestion in the mouth, and it breaks down the 1 and 6. Okay, 1,6 branch chain of the starch, and it breaks down and forms a chain. Then that food molecules with the help of tongue, which will be swallowed. Okay, which will be swallowed, and which is entered to esophagus. Okay, this is occur in the mouth. In the mouth, what happens? The mastication process. During the mastication process, that is chewing and grinding, the salivary amylase which comes into the contact with the food particles and break down the starch. Basically, rice is what? Starch. So that starch will be broken down, the bond, that is 1,6 bond, and that will be broken down and sent to the esophagus. That food particles enters to esophagus. As soon as it comes into the stomach, what is this organ? Stomach. When it comes to the stomach, the salivary alpha amylase activity will be less. That is, it will be nullified. It will be inactivated. Why? Because here in the stomach, the hydrochloric acid will be high. The production of the hydrochloric acid will be high. The pH of the salivary amylase is what? That is, the pH is 7, but the stomach's pH is 1 to 2. Okay, 1 to 2 is the pH of the stomach, but the salivary amylase is 7. So I told that the factors affect in the enzyme's activity. When it reaches to the stomach, it will nullify the activity of salivary amylase. So here there is no digestion occurs in the stomach. What happens here? When it reaches to the stomach, there is no digestion occurs. Then what happens? When it enters to the pancreas, okay, when it enters the pancreas or small intestine, first it enters to the pancreas, there will be a enzyme called pancreatic amylase. What is that? Pancreatic amylase. This pancreatic amylase has an action which breaks down the bond, that is alpha 1,4 linkage, that is alpha 1,4 glycosidic bond, and makes into some subunits. That is, some subunits maybe, that is glucose and disaccharides, the polysaccharides which will be break down into disaccharides and monosaccharides. Okay, then as soon as it enters to the small intestine, okay, exactly in the small intestine only the proper digestion that is occurs. Okay, the complete digestion that is occurs in the small intestine only. Here there are various of enzymes which will be produced by small intestine, and hence and proper digestion occurs by the small intestine. This is how the digestion takes place. First, when you ingest the food particles in the mouth, there will be a salivary amylase which comes into the contact with the food particles and break down some bond, and when you, with the help of the tongue, when you swallow it, that enters to the esophagus. When it enters to the esophagus, by the help of GI tract, that is gastrointestinal tract and also some hormone. It pushes down to the stomach. There is no digestion occurs in the stomach. Why? Because the HCl production, because of the high pH, the salivary amylase will be inactivated. Then it pushes to pancreas. Their pancreatic amylase, the pancreatic amylase which comes into the action, then the bond 1,4-alpha-1,4 linkage will be breakdown. Then it will be entered to the small intestine. As soon as the food enters to the small intestine, the proper digestion occurs and this completes the digestion. Okay, the digestion process will be completed. So can you say now what is digestion? Digestion is nothing but the breakdown of larger components, or the complex molecules will be splitted into a smaller components. That you call it as digestion. Digestion is nothing but the process of breakdown of larger components or complex substances into a simpler units. You call it as digestion. Next, coming to the absorption. After the digestion, how the stomach or how the small intestine can absorb the proper nutrients and supply to the wherever it is necessary. You can observe here. This is the structure of small intestine as well as stomach. Here you have sucrose, maltose, when the food products which is entered, and you have various of enzymes which is present in the brush borders. This is, you call it as intestinal lumen. In the intestinal lumen you have, for example, consider this is the intestine. Okay, if it is the intestine, this structure, okay, this is like this. This is a structure called brush borders. That intestinal lumens, you have a brush borders. Okay, these brush borders which helps to absorb the nutrients. For example, in this small intestine you have various enzymes like sucrase, maltase, lactase, like this, galactase, like that. For particular food particles, you have a particular enzyme. For example, sucrose, you have sucrase, maltose, maltase, lactose, lactase, likewise, that will be breakdown and forms a monosaccharide units. So you can observe here. What happens? You have some transporters. Okay, transporters, that is special glucose transporter is there, GLUT2 and GLUT1. There are many glucose transporters which will transport the glucose from inside. This is the cytosol of the intestine and this is the outside of the intestinal lumen. So what happens here? When the digestion is completed, there the intestinal lumen, these intestinal lumen, there will be a brush border enzymes. Those will be absorbed. Okay, what happens here? Absorbing by the help of an special glucose transporter, as well as you have some sodium and sodium potassium dependent voltage. Okay, these are the things which will be absorbs the carbohydrates. Okay, when for example here sodium will be influxed with the polarized. Okay, with the polarized it will be carries inside, and the wherever the potassium will be polarized into outside. Okay, then the glucose transporter which transport the specific glucose and fructose. Okay, for example, you know that in the polysaccharides we have many monosaccharides. What do you mean by polysaccharide? Polysaccharide is nothing but the sequence or the chain of more than 20 units of monosaccharides. You call it as polysaccharide. So those monosaccharides will be for particular transporter. These glucose transporter 5 or glucose transporter 2. These glucose transporter 5 only transports the fructose. Okay, and these glucose transporter 2 that transports only glucose molecules, and galactose or glucose, it will be transported from outside of the lumen into the inside, and then it will transport to the portal system, that is blood portal system. Okay, then when it enters to the blood, that blood carries and wherever it is necessary. For example, for specific liver, for liver it is necessary, the glucose will be transported to liver. If it is necessary to kidney, it will transport to kidney. If it is necessary to other tissues or other cells, it will transport by the portal system. This is how the absorption takes place, and you have lots of mechanism includes here, but as this is a pre-classes, so if you want that in detail, you can check our website. So next, if the enzymes is not working properly, what happens? How the digestion and absorption takes place? Is there any connections between enzymes and digestion? You got now. There is a connection between enzymes and digestion. So we know that lactose intolerance, this is a condition where the people, if they intake the lactose, they feel some allergic condition or they feel uncomfortable, right? This is the lactose intolerance. Where you can found the lactose? Lactose you can found in the milk. Where you can found? You can found in the milk. So lactose is also known as the milk sugar. So can we define this? Lactose intolerance, it is nothing but it is a condition that makes hard to digest the sugar in milk and milk products. If the product which is made up of milk and also the milk sugar, that is called lactose, where the lactose is indigested or we cannot digest the lactose if the absence of lactase enzyme. Which is that enzyme? Lactase enzyme. Okay, this lactase enzyme which is present in the small intestine, if this lactase enzyme is insufficient, it is not producing sufficiently, or this lactase enzyme it is lacking in the individual, then this lactose intolerance can be identified. Okay, so what do you mean by lactose intolerance? Lactose intolerance is the condition where exactly the lactose is indigested. It cannot be broken down into smaller particles. Okay, so what are the symptoms you can identify? By bloating and abdominal pain. If you intake that lactose, you can get abdominal pain and also bloating. Okay, and diarrhea, loose motions and excessive gas. Okay, in the small intestine only the excessive gases will be produced, and nausea and also reflux, that is the food particles will be reflux, that is vomiting. Sometimes it occurs vomiting. So you can see here the structure. In the small intestine you have a lactase. When you intake this milk, if the lactase enzyme is lacking in an individual, they shouldn't take the lactose or the milk products. Okay, then if it is occur, the bloating will occur, cramps, diarrhea, everything you can see here. These all are the symptoms you can identify by lactose intolerance. And also causes, you can see causes. How can you there are primary causes and secondary causes. Primary causes is nothing but actually insufficient. Okay, there is a insufficient of producing the enzyme lactase. Secondary lactose intolerance means where exactly the small intestine will be damaged. Okay, the damage of small intestine while operations or sometimes like that. If that conditions, the lactase enzymes will be not produced. And also genetically or hereditarily, the lactase enzyme cannot be produced. Okay, that is also the causes. And coming to the treatment, how can you come across this? Okay, the treatment is, first is dietary adjustment. You have to adjust your diets. Okay, for example, if you're consuming the milk, you should avoid. You have lactose free milk. Okay, in the market you will get lactose free milk. You can consume that. And also you should avoid the bakery products which is made up of milk. Okay, then you can reduce the risk of lactose intolerance, and also you can limit the dairy products. Then it is lactase supplements. Okay, lactase supplements using enzymes. For example, the lactase supplements you can intake like tablets. You can intake through tablets and or drops before consuming dairy. Okay, if you want to consume the dairy products, first you have to take that lactase intolerance tablets, then you can consume the products. Okay, that is also one of the management you can do. And lactose free products, as I told, lactose free products you can get in market, that is lactose free milk and lactose free food products you will get. Okay, that is you using milk or dairy substitutes. There is some alternatives, okay, that you can use. For example, soya, almond milk, soya milk. You can consume these type of things, and also you can manage the lactose intolerance. By this you can know that why enzymes are very important. Understood? And if the enzymes is not present, and if the enzymes are not produced properly, that may leads to various disorders, and also it leads to malabsorption also, and various disorders. Okay, this is about the enzymes chapter. So if you need in detail and also to understand more about various aspects of enzymes and also the procedures and whatever it may be, everything you can come across our website, that is Easiness website, that you can access through Play Store, and also you can access through the App Store. Okay, and we have a discount. You can see the screen on the screen. You can see that is 1500. It is offer, and you can also contact our Easiness, and also through the YouTube and email you can access our channels. And also it is very useful for PBBSc students and GNM students and also BSc nursing student. If you want to learn easily and you have to understand with the 3D animations, you can access our Easiness. And if you have any doubts, please comments in our comment section.